Hello viewers, I'm your mad buddy Arun and I welcome you to my first series of lectures on diseases caused by gram-positive bacteria. For this, my first lecture is on pneumococcal infections. In this lecture, I will take you through the history, microbiology, virulence factors, epidemiology, pathogenesis, clinical signs, symptoms, diagnosis and treatment of pneumococcal meningitis, pneumococcal pneumonia, otitis media and sinusitis. So let's get started. History. Louis Pasteur and George Sternberg in 1886 discovered for the very first time the micrococci from the blood of rabbit, which they inoculated with saliva. It was a diplococci. But by this time there were no reports of any harm from this bacteria. But by the end of 1930, in countries like USA and Europe, this bacteria was the third largest cause of deaths. By the end of 1974, Streptococcus pneumonia was renamed. Microbiology. Streptococcus pneumonia is a gram-positive, capsulated and chain-forming bacteria, while Diplococcus pneumonia is a gram-positive, capsulated but pair-forming bacteria. The polysaccharide capsule is important for us in serology. By using polyclonal antisera in past that we extracted from rabbit's blood, we have divided them into serological groups. Presently, there are 98 serotypes and 21 serogroups. Based on capsule-specific monoclonal antibody, their genetic variability and biochemistry, these serogroups and serotypes are formed. It is best cultured in presence of 5% CO2 and a catalase source. The catalase that we use comes from the blood agar. It forms green colored colonies, and this green color comes on reduction of iron that is present in hemoglobin. It is an alpha hemolysis, optigene sensitive and bile soluble bacteria. Virulence factors. It has a polysaccharide capsule which guards it from phagocytosis and opsonization. The same capsule also has pneumococcal H inhibiting factor, choline binding factor A, pneumococcal surface proteins, which altogether inhibits C3 formation in complement pathway. It forms biofilms, which is responsible for upper respiratory tract infections and otitis media. It produces neuraminidase, also called as sialidase, which breaks sialic acid and proteins in host cell. Pathogenesis. Dynamic nature of its capsule makes it capable of being colonized in the nasopharynx, causing non-invasive infections such as otitis media, sinusitis by down-regulating the capsule. By upregulating its capsule, it causes invasive infections such as meningitis and bacteremia. Immune factors such as TLR2 and 4, interleukin-1 cytokines, C-reactive protein and macrophages. These factors provide us innate immunity against this bacteria. Cystic fibrosis, bronchiectasis, COPDs, reduction in immune system, and smoking, and co-infection with a virus increases chances of invasive infection. The co-infection with viral increases cytokine release. In response to cytokines, these bacteria start producing neuraminidase, which damage the local lining. Peptidoglycans and teichoic acid which helps it in invasion, and capsule upregulation, which saves it from phagocytosis, and these all things increases chances of invasiveness. In adults, against this bacteria, adults have T-cell independent activation of B cells and release of antibodies, which saves time and makes them less susceptible to the invasiveness of this bacteria. While in infants, the immunity at this time is T-cell dependent, and formation of B cell and antibodies is T-cell dependent, which consumes time and makes infants more susceptible to the invasiveness of this bacteria. Mucociliary movement and immunoglobulin G prevents us from invasiveness of this bacteria. Most of the invasiveness or the spread of this bacteria to any of our organ is mainly by hematogenous route. Once inside the body, the capsular peptidoglycan and teichoic acid triggers the release of cytokines such as interleukin-1 and interleukin-6, which further increases the number of polymorphonuclear leukocytes and causes inflammation, and also TNF-alpha, which causes activation of alternate complement pathway. Epidemiology. It is globally present. It is an intermittent inhabitant of human's nasopharynx from very early age. In developed countries, up to 50% of children below 1 year of age get colonized by this bacteria. And in developing countries, this number is up to 90%. Yes, they live in our nasopharynx without causing any harm, but whenever they get chance, they don't leave it. Different serotypes of this bacteria have different number of infections based on age, sex, ethnicity, and geographical location. The 7 main serotypes that caused 70% of total infections in 1999 are 6A, 6B, 14, 19F, 23F, 1 and 5. The serotype 1 and 5 is omnipotent. It is globally present. It is present even in Europe and Sub-Saharan Africa. After the emergence of PCV vaccine, this number has reduced up to a considerable mark. The PCV vaccine, polysaccharide protein conjugate vaccine, has 59% serotype, has its effect on 59% serotype in infants, up to 70% serotypes in 18 to 64 years, and 57% in above 65 year